Welcome back, everyone. Here we are today for the MetCart lab. This is one of the labs, this is probably the lab that is the most valuable to do on campus. So of all the labs that we're having to do in this online format, this is most definitely the one that I'm saddest that we can't be there in person. We do have a metabolic cart on campus. It's really cool because we get to put one of you on the treadmill, and then we get to put the respiratory apparatus on that person, and then we get to have that person walk a mile and run a mile, and we get all sorts of fun data. So this is probably the one that I'm saddest that we're not be able to do it in person. But the situation being what it is, we're going to make do. So as you're going to see, we have some sample data from a couple years ago of someone who actually did do the lab on campus. So you'll still be able to do the equations. Okay, so here's our outline for this video. Just like we would if we were on campus, we'd be reviewing some things before we actually did the lab. So we're going to review the energy pathways during exercise. We're going to talk about ways in which you can measure calorie expenditure, and then we'll get into the MetCart, and then we'll use our sample data. Off we go. Okay. So this comes right from the lab. Let me use blue today. And how about red? I think red will go better. So this comes right from the lab book, and I know it is something that Dr. Thompson has talked about. So here we go. If we're asking muscles to exercise, there's a lot of things that they need in order to be able to contract. One thing that our muscles typically need is oxygen. But there's something else that our muscles need, and that is fuel. And in the case of muscles, the fuel source is ATP, adenosine triphosphate. Think about a car. If we want a car to be able to go, we've got to put gas in the tank. It's the same way with muscles. If we want to ask muscles to contract during exercise, they need fuel, and the fuel source is ATP. So what we're talking about here are the energy pathways through which our muscles generate ATP. These are the ways in which our muscles generate fuel that they need in order to contract. And we're going to be going in order. So what is it that our muscles use first to generate fuel? That would be the phosphocreatine or PCR system. This is what's going to provide fuel to our muscles for the first maybe 10 or 15 seconds of exercise. How it works is not really a big concern at this level of a course. It's a bunch of chemical reactions that are able to regenerate ATP within the muscle. But I do want to point out that this is anaerobic, which means it does not require oxygen. Okay, so what are the things to review? We're talking about ways in which our muscles generate fuel, which is ATP, and the way that our muscles first generate fuel. So when you immediately start exercise for the first 10 or 15 seconds, your muscles are generating fuel through a chemical pathway called the phosphocreatine or PCR system. This chemical system is going to generate fuel, ATP, for the first 10 or 15 seconds of exercise, and it does not require oxygen. All right, great. Now, what if you continue to exercise more than 10 or 15 seconds? Well, then you're going to start to utilize a second series of chemical reactions called anaerobic glycolysis. This is going to provide fuel in the form of ATP for an additional 30 seconds to 2 minutes. And this is also anaerobic, does not require oxygen. How it works technically is it converts glucose through glycolysis into ATP. But our biggest concern is it's anaerobic, it's what our muscles use second, and it provides fuel, ATP, for an additional 30 seconds to 2 minutes. And then, what if you continue to exercise past 2 minutes and 15 seconds or so? Then we're going to be able to rely on aerobic metabolism. And as the name suggests, this is when we can produce fuel, ATP, using oxygen. And because we finally have oxygen down to the muscles, this is our biggest energy source. We get our biggest bang for the buck. Our first two systems are anaerobic. They don't require oxygen. And then, as I just said, this one is aerobic. It does require oxygen. Our muscles would love to be able to create energy using aerobic metabolism all the time, but it's just not possible. And we can think about this. It takes time to signal our brain to tell our respiratory muscles to increase respiratory rate. Then we gotta bring in more oxygen. That oxygen has to travel all the way down your airways. Then it has to be exchanged between your lungs and the bloodstream. Then we have to transport that oxygenated blood all the way to the muscles. That takes time. So because it takes time to get additional oxygen to the muscle when we're exercising, we have to have means that first are anaerobic. We don't get as much fuel from them, but it's what we can use immediately in the short term. So cool. Okay. A couple more things to point out. As I introduced it in order. So when we initiate exercise, we're first utilizing the phosphocreatine. Then, as we continue to exercise, we're utilizing the anaerobic glycolysis. And then, as we continue to exercise, we're going to be using aerobic metabolism. So at the onset of exercise, we do in fact generate fuel in that order. It's always going to be first from the phosphocreatine, then from the anaerobic glycolysis, and then from the aerobic metabolism. But as you can see on the right, I have an arrow going up and down. Because after the initial onset of activity, if you've been exercising for a period of time, sometimes we do in fact go backwards. And then we may come back up this way. If we want to give an example, let's say that we're out for a run. Excuse me. You're out for a run, and at the initial onset of your run, you're going to be using energy from the phosphocreatine system for the first 10 or 15 seconds. Then you keep running longer, and then you start to use anaerobic glycolysis to produce ATP. And then lastly, you're going to be producing aerobic metabolism, which is great. But what happens if you come up to a hill? And all of a sudden, in order to propel yourself up that hill, you're going to need to increase the intensity of your effort. And you're going to need more fuel all of a sudden. Well, it may not happen that you have enough oxygen right there to increase the intensity enough to provide that fuel. So if you reach that hill and you have a sudden burst of intensity, you may in fact work backwards and for a period of time be using anaerobic metabolism. But then as you get to the top of the hill and you can lower your intensity a bit, then you may go back to the aerobic. Okay? Let me say that again just to make sure we're clear. At the immediate onset of activity, you're always going to be going in this direction. My arrow drawing skills are not very good today. At the onset of activity, you're always going to be going from phosphocreatine to anaerobic glycolysis to the aerobic metabolism. However, if you're doing some sort of continuous exercise and your intensity levels change, you may in fact start to work back into anaerobic metabolism for periods of time, only to go back again to aerobic. So that's why I have this arrow. I'm going to erase these messy arrows. Not my best work today, but that's okay. All right. Last thing I want to point out here is we can see the types of activities that will rely more on the anaerobic metabolism versus the type of activities that will rely more on the aerobic metabolism. So up top here, next to the picture of Usain Bolt, he's a sprinter, right? So at very high intensity activity, like a sprint, Usain Bolt is known for his 100-meter sprint. He did the 200-meter as well. What was his 100-meter world record time? 9.5 seconds or something like that. So at 9.5 seconds, he's obviously utilizing the phosphocreatine system. So what I'm saying is, in activities like sprints, where they're really high in intensity, but because they're so high in intensity, you cannot maintain a very long duration, you're going to be utilizing more of the anaerobic means, the phosphocreatine and the anaerobic glycolysis. Versus if we look at a marathon runner, this is a gentleman that just beat the sub 2 hour marathon, Kipchoge, last year. If you're working more in the aerobic metabolism, like a marathoner, he's obviously running at a lower intensity compared to Usain Bolt, but because he's at a lower intensity, he's able to hold onto that exercise for a longer duration. So when you're exercising at a lower intensity, but for a longer duration, then you're going to be relying more on the aerobic metabolism. All right. Okay, I know you love charts. We love charts. Okay, this chart has come directly from your lab manual. I just added some colors to make it a little bit easier. This is showing us exactly what we just talked about. On the x-axis is time of exercise. And on the y-axis is a percentage of the 3 energy systems that you're utilizing. Look at the onset of exercise. So at the very beginning of exercise, you can see that we're utilizing most of the red, which is our phosphocreatine or PCR system. So if I look at, 10 seconds of exercise, and I follow that up, I can see that I'm using over 95% of my energy is coming from the phosphocreatine system. But why I like this chart is because it shows us something else. We know the order that we go through to provide fuel to the muscles. But what this graph is showing us is that we're typically always utilizing some kind of combination of the 3. So I know that I introduced them in order of how we use them, 1, 2, 3, but there is some crossover. In other words, it's not like we utilize the PCR only, and then after 15 seconds, we switch to all anaerobic and nothing else. Instead, it's more of a gradual change, okay? And at a given time in exercise, we're probably utilizing some of both, or maybe even all 3. But the same general rule applies that at first, we're utilizing the PCR. Then, after about 30 seconds to 2 minutes, we're going to be utilizing more of the anaerobic, and then we're using aerobic. Let's look at a different timeline. Let's look at, about 30 seconds. At 30 seconds, if I were to draw this up, I'm utilizing probably the majority of the blue. So I'm utilizing 65% of the anaerobic means, and then 20% of the PCR still. Okay? And then, of course, as we get further along in our duration, past 2 minutes, now we're really going to be utilizing the most of the aerobic metabolism. Excellent. Good stuff. All right. Okay, so this lab is talking about assessing energy expenditure. And I want to make a point because this can be a little confusing. we can talk about energy in, and we can talk about energy out or energy expended. We are going to be focusing in this lab on energy out. And what energy out means is calories burned through exercise. Because that's our question today. How do we measure how many calories someone burns when they're exercising? But I want to point out that we've already covered energy in. And ideally, when you talked with Dr. Thompson about the energy balance or the weight management chapter, ideally, you want to match energy in and energy out. Let's review. Energy in was calories you bring into your body through food. We've already talked about this. We had a couple labs on it, as a matter of fact. Didn't we talk about how many calories MyFitnessPal recommends that you ingest through food? Well, of course. So we've already talked about energy in. How many calories you should have based on your age, height, gender, activity level. We even measured the amount of calories in food, didn't we? We put the food in either a homemade or a bomb calorimeter, measured how much heat comes off of it. So we've already covered this side of the equation, calories in through food. We also can assess calories out, how many calories we burn every day. How many calories our body burns every day. And that's going to be our discussion today. Energy out, energy expenditure, calories burned through exercise. Okay, these words are going to look familiar to you because guess what? When we looked at energy in, when we looked at the energy content of foods, we used indirect calorimetry because we said direct calorimetry is too hard. Well, guess what? When we measure energy expenditure, calories burned through exercise, it's the same story. It's possible to do direct calorimetry. Look at that top picture on the right. You have a dude exercising in a sealed little room, and you have water flowing through the top of the room, and then we can measure how much hotter that water gets as the person exercises. But that's really difficult. I've never seen that done. I've never known of anybody who's done it. So I would say it's theoretically possible, but it's not very practical at all. So instead, we utilized an indirect calorimetry, an indirect way of measuring how many calories someone burns through exercise. In this lab, we're going to be talking about two ways that we can measure energy expenditure through indirect calorimetry. Accelerometry, and then our good old friend, the metabolic cart. So I just want to mention accelerometry. We do not do this in our lab because we do not have the equipment, but I think it's useful to discuss. This is an indirect means, and an accelerometer, what it does is it measures the acceleration and the positional changes of the body during exercise. There's different types of accelerometers. A couple years ago, the Nike Fuel Belt was really popular. You can see it's like a little bracelet. You can attach a calorimeter to someone's ankle, and you can see the cal track there. No matter the type of accelerometer, it has the technology that is attempting to measure positional changes. So the more you exercise, the more intensely you exercise, you're going to have greater positional changes over a shorter period of time. And based on your acceleration and your positional changes, the unit puts that information into a formula. And of course, based on your age, height, weight, gender, activity level, it will estimate energy expenditure. Is accelerometry a great way to do it? Well, what are the pros? I mean, it's obviously convenient. It's much cheaper than the Metcart. The CalTrack unit, last time I looked, was like $45. Of course, the Nike Fuel Belt, they're going to upcharge you 100 or so. So it's easier to use and it's more practical. It's cheaper. But on the downsides is its accuracy. So I've given you some references here that show you the accuracy of this is perhaps not great. So while it's convenient and cheaper, it is not nearly as accurate. All right. In this lab, we're going to be using the metabolic cart. You can see it on the bottom right. That looks very much like what we have in the lab on campus. There's a treadmill, and then there's a little cart that has a computer and the actual metabolic system. Make sure you review how the metabolic cart, maybe I should add a title to this. I didn't even add a title. The previous slide was accelerometry. This is our metabolic cart. All right. Make sure you review how this is a means of indirectly measuring energy expenditure. So as you can see on the picture here, and there I am on the top, but look on the bottom picture, you can see attached to the unit are these gas canisters. One of them is feeding oxygen to the person. And then the little apparatus here, let me circle it on myself. The apparatus is a two-way non-rebreather. So it's able to separate the oxygenated air that comes in. So we can quantify exactly how much oxygen you're bringing in. And then we're able, because it's a two-way non-rebreather, separate the air that's coming out. Because you want to see how much CO2 you're breathing out. And if we were to follow the out tube, it eventually goes to something called a mixing chamber. And that little box unit is able to analyze the air exhaled. And from that, from the gases exchanged, from the amount of oxygen the exerciser brings in, and from the amount of carbon dioxide the exerciser breathes out, we're able to estimate energy expenditure. Why? Because the amount of gases you bring in and out is relative to the amount of fuel used by the muscle. In other words, at a lower intensity activity, your body doesn't need as much fuel. It needs some, but because it's a lower intensity or lower level activity, you don't need as much fuel. And if you don't need as much fuel, you don't bring in as much oxygen and you don't produce as much CO2. Versus a high intensity activity, your muscles need more fuel. And because your muscles need more fuel, you bring in more oxygen and you exhale more carbon dioxide. So because of that relationship, we're able to take the gas data, the amount of gases you exchange, and we're able to estimate how much fuel your muscles used, and therefore able to estimate how many calories you burned as a person. Pretty cool. I really wish you could see it. All right, now here's the med cart, and I've already kind of talked to you a little bit about it. We can see that there's a gas chamber. Oh, let me actually hit the button here. There's a little gas chamber. There's actually two of them. Talked about this is a two-way non-rebreather, so it separates, so we can quantify the amount of air coming in, and we can quantify the amount of air coming out. The amount of air coming out goes into a little mixing chamber that's able to analyze that air, and then come up with some estimates of how many calories we burned. I do want to point out, even though it's kind of hard to see here, this mask is covering her nose, right? Yeah. And if I go to, there's a picture later on. All right, I'm not going to worry about it now. There's a picture later on, even on mine, When I did this, I had to have a nose clip over my nose. So let me ask you this. Why is it important that we cover the nose or we close it? Well, the answer is because we want to be able to quantify all the air coming in and coming out. And if I have the nose open, then you're automatically going to be breathing in some air and breathing out some air through your nose. And if you're breathing in and out air through your nose, we're not quantifying it, we're not capturing all that gas, and therefore our measurement will be inaccurate. So we want to make sure we close the nose off so that we can only breathe through our mouth, and you're able therefore to capture all the air. Okay, one more thing before I go on, because you've probably heard about this. This equipment, the Metabolic cart, is commonly used to do a max VO2 test. We do not do this in the lab because it's a bit above the level of this intro course. It's also a pretty challenging test. So you don't have to know this for the exam here in lab, but I just want to point it out because you may have heard of this equipment not for measuring how many calories someone burns, even though it's possible, but you may have heard of this equipment because it's a way of measuring max VO2. Max VO2 is a measure of aerobic fitness. So if you want to see how aerobically, physically fit someone is, this is the gold standard test. I've done it a couple times. How it works is you put someone on the treadmill, same little apparatus around their mouth, but instead of just having them walk or run at a steady pace, it is a graded protocol. It's really hard, let me tell you. You start off with the treadmill being flat, and you start off at a pretty low speed of running. But then, every 2 minutes, the speed of the treadmill increases, and the grade, the incline of the treadmill increases. The goal is for the person to keep running as long as they can. Imagine, at first, I've done this a couple of times. At first, it's no problem, out for a jog. Even the first time the speed goes up and the grade goes up, you're thinking, oh, it's just a little hill, I got it. But every 2 minutes, the speed and the grade goes up. You are working hard very quickly. The longer you can exercise, usually the more physically fit you are, but the longer you can exercise on this protocol also gives us a more accurate reading. And what it's trying to assess is at your max. So ideally, the person would keep running until ultimate failure. That's difficult to do because usually our nervous system shuts us down before we reach complete failure. But the idea for this test is to estimate at complete failure, what's the maximal volume of air you're able to bring in? Because the more physically fit you are, the more aerobically fit you are, the larger volume of air you can bring in. All right, so you may have heard of the Met cart because of the max VO2 test. We're not doing it in the lab, just wanted to touch upon it. Here's what we are doing in the lab though. In the lab, instead of doing a max VO2 test, we're going to assess the number of calories a subject burns walking a mile, and then also running a mile. So instead of doing the max VO2 test, we can utilize the same equipment, we can understand the same equipment, and we can estimate the number of calories someone burns walking a mile and running a mile. That's what we're going to do. Okay, some terms. I already talked about VO2. Just make sure we can define it. VO2 is oxygen uptake, or the volume of oxygen someone brings in. Then we have the volume of carbon dioxide produced as they exhale, the VCO2. This Met cart also can give us the RER, which we already know about. And wait a minute, you know what the respiratory exchange ratio is? How do we find it? VCO2 divided by VO2. So it is precisely this gas data that we just do simple division to give us the RER, which, as we know, tells us what nutrients our muscles are burning for energy. Lastly, this unit also gives us an estimate of resting metabolic rate. This may be a new term for you. You may hear it called basal metabolic rate. Either way, basal metabolic rate or resting metabolic rate. This refers to the amount of energy expended by the body at rest. In other words, not exercising, not walking to the refrigerator, how many calories your body burns just to keep its vital organs functioning. So if you were to sit on the couch all day, not that you would ever do that, but if someone was to sit on the couch all day, their body is still burning calories because it takes energy to keep the heart pumping, to keep the liver filtering blood, to keep all of our organs working. You might be surprised at how many calories it takes just to keep our basal activities alive. I'm going to change color here. There's a quick and dirty way that you can think about how many calories you burn just to keep your vital organs functioning. If you take your weight in pounds and you multiply that by 10, that's a really broad, quick and dirty way of estimating it. For example, if you have someone who's 150 pounds, if I multiply that by 10, which just means move the decimal place over 1, that would be 1,500 calories. Now this is very broad. We're going to do a better calculation in a moment. But if you just ever wanted to get really quick and dirty, take someone's weight times 10. So that means that for a 150-pound person, their body burns 1,500 calories just to keep their vital organs functioning. And then calories burned through exercise gets added onto that. Now, I just mentioned it. We did the quick and dirty way up top with body weight times 10. But you are going to do a more specific formula. This is right from your lab book. Make sure you choose the right formula to get a calculation of basal or resting metabolic rate, one for females, one for males. In order to do that, you're going to need your height in inches and your weight in kilograms. Okay, so here are the conversions for that. To do height in inches, take your height in inches. So, for example, if someone's 5'8", they're 68 inches. 5 feet is 60 inches. So if you're 5'8", 68 inches. If you're 5'5", 65 inches. So you would take the number of inches times 2.54. This person would be about 173 centimeters. Take your weight in pounds. Someone's 152 pounds. We divide by 2.2 and we get 69 kilos or kilograms. So you'll need to do that for yourself. Feel free to pause the video to do that because you'll need these numbers. Moving on, you'll plug those numbers in. Okay? Make sure that you do the things in parentheses first. So you can see I did this for myself. I'm a female. I inputted my weight in kilograms. I inputted my height in centimeters. And then you're also going to input your age. Do the things in parentheses first. So I did 9.56 times 69, and I inputted that number here. Then I did 1.85 times 149. I inputted that number here. Lastly, I did 4.68 times my age, and I got this. Then you can go all the way across from left to right. For me, my basal metabolic rate, in other words, how many calories are burned by my body just to keep the vital organs functioning, is about 1,400. How does that compare to weight times 10? This was about 1,500. This was about 1,403, pretty close. Basal metabolic rate is largely determined by genetics. However, there are some things that influence it. One of them, and the reason why this one is lower for me, is age. As we get older, our basal metabolic rate goes down. Gender plays a role. Females tend to have lower metabolic rate than males, for example. Okay, now guess what? We can take that number, and here's another way that we can use a formula to estimate how many calories we need to intake through food. I realize we're talking mostly about calorie expenditure with exercise and basal metabolism in this lab, but it just so happens that here's an opportunity to show another way to assess how many calories you need through food. So you'll do this for yourself. You'll take the basal metabolic rate that you got. Mine was 1403.19. 1403.19. So you'll take whatever your basal metabolic number was, and you're going to multiply it by a guesstimate of physical activity level. This is a table that comes right from your lab book. If this were me, I exercise 5 or 6 days a week pretty intensely. I would say that would be for me. So I would take my basal metabolic rate times that. And this is another way of getting an estimate of how many calories you should be ingesting per day. I'm doing the math right now. 725. This is telling me I should be bringing in about 2,420 calories per day. It'll be interesting for you to see how this compares to what MyFitnessPal recommended for you. Just another way. Okay, here we are, finally, the MetCart Lab. If we were in person, we would have one of you who would volunteer to go on the treadmill. Because we're not here, I have some sample data of someone that we used in lab a couple years ago. This is a pseudonym to protect his anonymity, but the rest of the information is what it was. So we have a 24-year-old male. We're going to name him Johnny Football. 250 pounds, who exercised at a moderate activity level. The purposes of this metabolic cart lab: we're going to determine those gas values and RER using the metabolic cart, which is a form of indirect calorimetry. And then we're going to use the gas values to estimate the number of calories this subject burned walking 1 mile and running 1 mile. So I'm going to pause for a minute because now it's your turn to develop some hypotheses. So somewhere, go ahead and jot down how many calories you think this subject burned walking 1 mile, and how many calories you think Johnny Football burned running 1 mile. Okay, so if you had to pause the video for that, that's great. But now we're going to get back started and talk about how we ran the test. So here's Johnny Football on the treadmill. What we did is we first had him resting for 10 minutes. So we put that apparatus on his mouth and we had him sit down with the respiratory apparatus on for 10 minutes. Can you think about maybe why we would do that? Well, the reason is because we want to make sure that his resting values are really resting values. Because what happens, you can imagine this, if you're in a lab and you're going to be the subject, you're probably nervous or excited. Your heart rate's probably going to be elevated. Maybe you just ran across campus to come to lab to get there on time. So if we just started the walk right away, his resting values might not actually be resting. They're probably a little bit elevated from resting. So you always want to start by having your subject in a seated position for a period of time to normalize all the data. And of course, as an aside, this is an opportunity for you to make sure that the apparatus is working properly. So after a 10-minute seated rest period, we had Johnny Football stand up, and at his own pace, however fast he wanted to, we had him walk for 1 mile. And we collected the data every minute as he walked for that 1 mile. After he completed a walk for 1 mile, we had him move straight into a run for 1 mile on the treadmill at whatever pace he felt comfortable. Lastly, we had him do a cool down by walking on the treadmill at a slow pace for 5 minutes afterwards. We wanted to see not only how his gas values responded to the exercise session, but we also wanted to see how the gas values responded after he stopped, how quickly they returned to his resting values. All right, now, if we were in class, you would be helping us out, and we'd be assigning you duties. We would have someone as a student helper who would be our recorder, because we're going to be recording these gas values and RER and heart rate every minute. We would have someone assigned to be a headset adjuster. As you can see in the top right, that is very similar to the piece of equipment we have. You can see that the headset has some adjustment knobs, typically going from rest to walking and then walking to running. Sometimes it jostles around a bit, so certain parts of the headset need to be tightened. So it's good to have someone there to help with that. We would have someone be a treadmill operator so the subject can focus only on the walk and the run. And then check out the bottom two. We would have someone be a paper towel provider. With this apparatus in your mouth, sometimes some spit can kind of escape and you can have some drool around the mouth. So we can have someone wearing gloves, of course, provide paper towels to the subject to wipe that off. And in some instances, we would need to replace the drool cup. Now, you can bet I didn't call it the drool cup replacer. I used a much nicer term, so someone would actually volunteer. And then you can imagine their surprise when they found out what their job was. We provided gloves, though, to be fair. You can see coming down from the apparatus is this little vessel. And the job of that vessel is to collect spit. It is kind of an awkward thing having this thing in your mouth. So some subjects tend to be heavy droolers. Some subjects tend to not be heavy droolers. But if we have a heavy drooler, sometimes halfway through we need to pause the experiment and have someone unscrew that little cup and replace it with a new one. All right, so we have our subject, and here is his resting data. This is real data, folks. Resting data. So, after 5 minutes of rest, our 24-year-old, 250-pound football player had a VO2 of 306 mils per minute. These are in mils. A VCO2 of 415 mils per minute. Heart rate was pretty high. Maybe he was nervous. These values may not mean much to you now, but I'd like you to think about how much you think they're going to change when we go to the walk and then when we go to the run. They're obviously going to go up. But I want you to kind of just think about it. How much do you think they're going to go up? I also want to point out here the RER. If we did the math and we did VCO2 divided by VO2, we actually get an RER that's greater than 1. I want to say it's like 1.2 or something. This is just one of the inherent things with the METCART. Sometimes we get values of RER greater than 1.2, but that's not really the physiological truth. So if it's ever over 1, we just estimate it down to 1.0. Think about what that means, right? So you got to think about, remember what the RER values are. At an RER of 1.0, what nutrient are his muscles using for energy? That's pretty high at rest, but our Johnny Football may have been just a little nervous. Okay, so after rest, we had him walk, and here is the walking data. Real data. So he walked 18 minutes, and every minute we took data. Look at how much the VO2 went up. What was it at rest? 306 mils? It went up to 1,900, 2,000. That's a lot. we're talking about easily a 4 to 5 fold increase. 5 to 6 fold, really. So what does that tell us? That tells us that at rest, our gas values are very low. Your body does not want to waste energy. So at rest, your body wants to exchange as little amount of gas as possible for what your body needs. So it keeps it very efficient, and it means that we don't waste energy at rest. What this also means, though, is when we go to exercise, we have a lot of room to increase, and that can be very helpful during exercise. I'm asking you some other questions here. Some of them I'm not going to give you the answer because I want you to think about it, and you will, of course, have some lab questions to answer. But one of the questions I ask here is, why is it that the VO2, and for that matter, the VCO2, why do they change? Sometimes it's as high as being over 2,000, then it goes down to 1,300, then back up to 1,900. So why is it that there's some natural variation? Because that is normal. Lastly, I want to address the RER. At the beginning of exercise, Johnny Football had very low RERs. And this was actually a little bit unusual. Well, I will tell you, though, and I actually made a note of this when we had Johnny Football in for the lab, he told us that he did not have breakfast. And this was a morning lab. I want to say he got on the treadmill about 9 a.m. So think about it. The RER was lower than it normally would have been at the start of walking. Think about it. Why would the fact that he didn't have breakfast mean that his RER values were lower than where we would expect them to be? Okay, now, once we get to the end of this lab, you're going to have to do some calculations. And to prepare for those calculations, you're going to have to do some math. So I'd like you to do this right now. Feel free to pause the video after I explain it. You're going to need the sum. So you're going to have to add up all the VO2s. Add up all 18 values and give me the sum of the VO2s. You're also going to need to do the sums of the VCO2. Lastly, you're going to find the average of the RER values. So you'll add up all the RER values and divide by 18, because there were 18 values. So pause the video, go ahead and do that. When you come back to join me, I'd like you to think about how these values are going to change. How will the VO2 and VCO2 change from walking to running? We talked about the VO2s being 1,900, 2,000. How is that going to change when he goes to run? How will the RERs change? Will the RERs go up or down when he's starting to run? To answer that question, you might want to consider what nutrients his muscles are going to need. So let's think about it. Going from walking to running, he's going to be increasing the intensity pretty significantly. So when you increase intensity, what is the fuel, carbs, fat, or protein that the muscles are going to utilize? And then once you can answer that question, then you can figure out what the RER is likely to be. Well, here's the running data. The VO2 and VCO2 went up a whole lot. We're close to 4,000 mils of oxygen brought in per minute. Sometimes it dipped down, sometimes it was in the middle between 3,000 and 4,000. That is huge. Remember our resting values for this subject? 306 mils of oxygen brought in per minute, 306 at rest. Then we did walking, 1,900. And now we're 3,900. And the VCO2 values went up as well. So remember, we keep our gas exchange values very low at rest because it's efficient. We don't want to waste energy. But it also gives us so much room to grow. We have an incredible capacity to bring in and breathe out gases. And that's all in the name of providing oxygen to the working muscles and then getting rid of the CO2 that is produced from the working muscles. And then have a look at the RER. How did these values compare to what was happening at the walk? And then why? Just like you did for the walking, you're going to have to do some math for the running data. So you're going to find the sums. You're going to add up all of the VO2s. You're going to add up all of the VCO2s. Then you're going to find the average RER. And of course, this time, you'll add up all the RERs and you'll divide by 10, because that was how many values we had. Here is his recovery data. This is actually 3 minutes after his run. This is not going to be part of any of the calculations, but I think it's worthwhile to look at. As we said, it's just as important to see how the numbers go up or how they change during exercise, but we also want to see how someone recovers. So how do these numbers compare to his resting values? Are they close? The more physically fit someone is, the more quickly they will recover to their resting values. Also have a look at the RER. We can tell from the point of running down back to walking slowly to recover, the RER went down so much. So why would that be? All right, now, here we are at the very end. You're going to be doing some calculations. These are the same formulas from the book. Let me walk you through them. There are 4 different formulas or things to do, A, B, C, D. Letter A here, you're going to be inputting the average RERs that you previously calculated. Now the RER at rest, there was just one value, and you may remember the RER at rest was 1.0. And then I'm going to have you fill in the RER that was your average for walking and the RER that was your average for running. Part B, once you recopy your average RERs, you're going to go to a chart, and we're going to find for that RER what was the exact percentage of carbohydrates being utilized by the muscle, and what was the exact percentage of lipids or fats being utilized as fuel for the muscle. I'll use the example of rest, and then you're going to do the walking and running. Here are the charts, copied from your lab book. Add an RER or RQ, because we generally tend to use RER and RQ interchangeably for our purposes. Add an RER of 1.0, because that's what it was during the rest. At an RER of 1.0, I look over here. At an RER of 1.0, the muscles are burning 100% carbohydrate fuel and 0% lipids, which we actually already knew. But for the other numbers, you'll need this chart to get the exact breakdown. So I would go back here and I would write down at an RER of 1.0, the muscles are burning 100% carbohydrates and 0% lipids. But you'll fill in walking and running. Now we get to part C. Part C, we're going to be utilizing the same chart, only this time we're going to be looking for the kilocalorie per liter. I already did sitting at rest, and we already know that at an RER of 1.0, you're burning 100% carbs and 0% lipids. But now, from the same chart we just used, I want to find the kilocalories per liter of oxygen. So this time, let's change colors. I'm still looking at the RER of 1.0, but now I want to look to see what was the kilocalorie per liter of oxygen. And for an RER of 1.0, 5.047. So I would write in 5.047. And you will do the same for walking and running. Now we have all the information we need to see how many calories this person burned. The first thing you'll do, this is part D formula. You're going to take your sum of the VO2 from the walk and your sum of the VO2 from the run, and we're going to convert it from milliliters to liters. And of course, we do that by dividing by 1,000. I'm going to throw in some numbers as an example, but these are not the actual sums because I haven't done the math yet. But let's say that our sum of the VO2 was 28,000. Again, that's not the actual value. You're going to have to do the math yourself. I'm just giving you an example. We would divide by 1,000. And that just means taking our decimal 1, 2, 3 places to the left, which means 28.0. Whatever your sum of VO2 from the run is, maybe it's 31,000. Again, you're going to write in the actual number. I would do the same thing. Move a decimal place over 1, 2, 3, and I get 31.0. These are not the real numbers. I'm just giving you an example. You're going to put in the numbers that you get. So you'll first take those sums of VO2 and you will divide by 1,000. Then we get to the cash cow. Here we go. So whatever your sum of VO2 was for the walk in liters, again, I'm just going to give you sample data. Let's say it was 28.0. And then your sum of the VO2 in liters from the run, maybe it was 31.0, whatever. You then take that sum of the VO2 in liters and you multiply it by the kilocalories per liter of oxygen. That was the number I have in blue here. So whatever your kcal per liter of oxygen was for walking, you're going to plug that in right here. You multiply the 28 or whatever your number is times that, and that's going to give you the number of calories burned during the 1 mile walk. We'll do the same thing for the run. All right, so finally you get to ask yourself some questions. Was there a big difference in how many calories were burned during the walk and the run? How would these numbers have changed if we didn't have a bigger football player? What if we had someone who was 150 pounds versus 250 pounds? How do you think that would have changed the number of calories burned during the 1 mile walk and 1 mile run? And of course, there's going to be more questions to come from the lab assignment. Sorry we couldn't be there in person, but this is real data. If you have any questions, as always, reach out to us.